Hey everyone, welcome back. So in our last video, we wrote a blazing fast two-line Python function to count K-mers, and we successfully chopped a DNA sequence into tiny words and mathematically profiled it. But there is a golden rule in data science. Nobody wants to read a giant text dictionary. If you hand your terminal output showing 5,000 different formers and their counts to someone else, someone who is working with you, someone who is your superior, they will not know what to do with that and they will probably not like it. So today we are going to fix that. We are going to extract the most important data from our K-mer dictionary and build a visual fingerprint using Matplotlib. So let's jump into VS Code and paint with our data. So first let's paste in the pro level K-mer function we wrote in previous video, right? And generate a dummy sequence. So let's imagine we are analyzing a highly repetitive genomic region from an agricultural crop, like a strain of rice. So let me copy those commands that we made last time. This is the import statement, okay? And we will also copy our pro function that we used last time. This is the that function. Okay. Now let's generate a 10,000 base sequence with a heavy uh, GC bias. Okay. So bases will be A, T, C, and G. Okay. Next we will assign weights to each of them. So let's suppose 0.15 for A and T. And since we want it to be have a GC bias, so we will increase that to 0.35 and 0.35. This will force more C's and G's, right? Now we will make crop DNA is equal to uh, dot join random choices. For random choices, we will use weights uh, is equal to weights and K will be 10,000, right? After that, we will count the three-mers. So we will write K-mer counts is equal to count K-mers pro function, right? We will put crop DNA in here and we will assign K is equal to 3. Okay, and finally, we will print a statement that will tell us total unique three-mers found, and we will simply print it. Okay, if we run this, it will tell us something like this. Okay, yes, we made a mistake here. Weight will be weights, and this is fine. So if we run here, you will see that total unique three-mers found is equal to 64. It tells us we found all 64 possible three-mers, but plotting 64 tiny bars on a graph is too noisy. So we just want to see the dominant patterns, right? So to make a clean graph, we usually only plot the top 10 or 20 most frequent K-mers. So because we use collections.counter, extracting this is incredibly easy. Okay, so what we will do here is top 10 is equal to K-mer counts dot most common, and we will use 10 for that. And finally, we will print top 10. So when you print top 10, notice the format. It is a list of tuples, right? So to plot this in Matplotlib, we need to split this into two separate lists. One list for the labels, which is the DNA text, and one list for the values, which is the numbers. We can do this using a quick list comprehension. So what we will do is we will split the tuple into X and Y. X will be for labels and Y will be for values. So let's do that. X labels is equal to item 0 for item in top 10, and similarly let's copy it and we will name it item values, and instead of 0 we will use 1 over here. And finally we can print f labels is equal to K-mer labels, and let's copy this, paste it, and instead of labels we will use values is equal to K-mer values, right? And here you have it. Now we have split it. Okay, so now that we have clean x and y lists, building the bar chart takes just a few lines of standard Matplotlib code. And since we have already imported Matplotlib, we can just go ahead and do that. So what we will do is plt dot figure, and let's assign the figure size. Let's use 10:6. After that, we will create the bar chart. So plt dot bar, we will use K-mer labels and K-mer values. For color, let's use suppose teal. That should be fine. And finally, for edge color, I'm doing this to show you so that it's more visually attractive after making it. Okay, we can just make the bar plot simply without using these colors, but still let's do it so that you can learn. So now we will assign the title. What will be the title? It will be top 10 most frequent 3-mers, right? Yeah, we are using 3-mers in crop sequence. Okay, let's assign font size as well. Font size is equal to 14, and font, I think that property is called font weights. I think we will check it and we will say bold. After that, we will use the label names plt dot label. That should be x label, right? And for x, we will assign K-mer sequence. Let's say font size is equal to 12, and similarly plt dot y label. Y label will be frequency, and font size will be 12. And finally we will let's do one more thing. We will polish the x-axis as well. If you use longer K-mers like 6-mers, the text will overlap. So rotating the labels 45 degrees is a crucial pro tip. Okay, so remember this part. So we will use x ticks, and rotation is equal to 45. And yeah, one more thing, we can add a subtle grid behind the bars so that you can actually understand the data. So let's use that as well since we are doing everything already. So axis y line style, we will use dash for that. And finally I think alpha, yeah, alpha is equal to 0.7. And what else? Okay, now we can just display the graph. So let's use a tight layout. It ensures that nothing gets cut off. And finally plt dot show, and look at that, we have a beautiful publication ready visualization. Because we forced a GC bias in our random data generator, you can clearly see that the dominant peaks are GC rich 3-mers like GCC, CCG, CGC and all that, and the 80 rich K-mers don't even make the top 10. And there you have it. You have taken basically raw DNA, chopped it into statistical chunks and rendered it as a visual fingerprint. If you compare the K-mer histogram of a human to the K-mer histogram of a virus, they will look completely different. And this is how you identify sequences without aligning them. And this concludes our deep dive into pure Python string manipulation and K-mers, and we have built an amazing toolkit. And as always, The code will be available in the GitHub repo, and I will give you a small challenge. You try to take the K value to 5 and see how the graph changes. And hit the like button, subscribe, and make sure you keep following the course, and I will see you in the next video.